Hi, my name is Heather McCarthy, and this is task number 8 for VETT 222. We will be doing a modified knots test today, which is a test looking for the microfilaria of heartworms. So what we're going to do is I'll show you everything I have set up really quick. I have my gloves on and I have my lab coat and my goggles. We are going to be working with formalin today, so we need to make sure that we are fully protected. So speaking of formalin, I'll go ahead and show you all of the materials that I have set up. So we have a tube holder set up. We do have 2 of these 15 milliliter conical centrifuge tubes. We have a 12 cc syringe that I'm going to be using to dilute out my formalin. I have my blood sample here. This is a freshly obtained lavender top tube. It is filled appropriately to the minimum, and so we have that ready to go. I have a 1 cc syringe that I'm going to be using to measure out my blood sample. I have a pipette, mixing cup. I do have some new methylene blue stain, and I have some formalin. This is 10% formalin, and then I have some distilled water. So the first thing that I need to do is I need to go ahead and dilute my formalin. So what we'll do, oh, sorry about the, oh my gosh, sorry about the vibration and everything. So what I have, the reason I have this cup out is this is just a sterile urine cup. I'm going to use this for diluting out my formalin. So I'm going to add in some distilled water. And then I'm going to draw up 8 milliliters of distilled water. And we'll make sure we get all the bubbles out just to make sure that we're measuring appropriately. So we've got 8 milliliters of distilled water. And then because this is sterile, I am going to go ahead and pour this back into this distilled water. This was sterile when I opened it. I've only put the water in there. And this is not going to be used like for anything other than lab procedures. So I'm going to place that to the side because I don't really need it anymore. The next thing I'm going to do is I'm going to add some formalin to my cup. Now we have 10% formalin, we need 2%, so I'm going to mix 2 milliliters of the formalin with my 8 milliliters of water, and that's going to give us the appropriate dilution. And I'm going to make sure that I don't splash this, and I'm going to try not to pour too much extra because I don't need that much. And this is a hazardous material, so. Alright, so then we're going to draw up 2 milliliters. And then I'm going to just let this mix inside of the syringe by just rocking it. And then I'm going to go ahead and put the lid on my cup just so I know that I don't spill it. This is going to be properly disposed of at the end of the this procedure, so I am going to sit it right with the formalin. I don't need to put a secondary label on it because it's going to be disposed of immediately after, and it was just mixed right now. So what we've got here is our dilution. Whoops. So we've got 10 milliliters here of our 2% formalin. We're going to go ahead and add this into our test tube. And then we're going to mix in 1 milliliter of our EDTA anticoagulated blood. I am going to go ahead and mix this well before we do that, just to make sure that we have a nice sample to put in there. I'm just going to gently invert it before I use the sample. And so now I'm going to go ahead and get that out. Like I said, we need 1 milliliter, so that's why I have this out ahead of time. So there's a little bubble at the end, but it's okay because I can see exactly where the line of the blood is. So just so you can see, that is 1 milliliter here of our blood. So we're going to go ahead and add this into our test tube as well. And this is a no hub loss syringe, so that's helpful to avoid losing your sample. So now that we have these together, we're going to go ahead and gently mix them. I'm going to do that by placing my thumb over the top of this firmly, and then we're just going to invert it several times, and this is going to thoroughly mix our sample. So it's very important that I'm wearing gloves right now because formalin is, it's caustic and it can cause damage to your skin. It is linked, I believe, with cancer as well, so that's not something that we want to get onto our skin. So this has been mixed up. The next thing we need to do is we need to centrifuge it. I'm going to go ahead and place some water in a tube just to balance it. And those are pretty close. So then I'm going to go ahead and spin my camera around so you can see the centrifuge. And we're going to place these into the centrifuge right across from each other because the centrifuge needs to be balanced. And we're just going to take these extra ones out because we don't need them right now. So we have our 2 tubes in here. They're balanced, and oh, you can see, but not quite as much as I should show you. So we're going to go ahead and close the lid, and then we're going to centrifuge this at 1,500 RPM for 5 minutes. So it's already set at 1,500, and then we just need to get the timer set to 5. And so we'll go ahead. I have some photos that we'll talk about, and we'll do that at the end. So I'll go ahead and read the slide, and then we'll talk about what we're looking for. So when we read our, sorry, my camera's being a little weird. When we read the slide, we're going to need to differentiate between the Dirofilaria imidis and another non-pathogenic nematode. So we will do that in more detail, and I have a chart to show the difference, how we distinguish between them. Right now I'm going to go ahead and put in a cutscene just because all we're doing is waiting for the centrifuge. So I will be back in about 5 minutes. Hi, this is Heather McCarthy, and I am back with part 2 of task number 8, which is the modified knots test. So our timer is about to go off for the centrifuge, and then we will continue with the test. So what we're going to do is we're going to decant the supernatant, which is basically just the fluid that is not sedimented down at the bottom of the tube, and then we will do our test with the sediment that's left. So our timer just went off. I'm just going to give the centrifuge a few moments to stop spinning completely. So we'll take a look and see. We have completely stopped spinning. So we're going to go ahead and take out our tube and check and see if we have anything at the bottom, which I don't see, but we'll see once we pour it out if there's anything. It looks like there is a little bit right there. So what we'll do, actually I'm going to go ahead and pour it out over here. So I'm going to just use the little cup that I had my formalin left over in, and that way we can dispose of this later all in one place. So what we're going to do is we're just going to pour off everything that did not centrifuge down. So you can see, it was hard to see in the tube before, but you can see that we have some sediment there that's not, it's not liquid, it's not moving and pouring off. So there is some sediment there, which is good because that's what we need for our test. So I'm just going to place that in there to make it a little bit easier. And then we're going to close this up again so we can dispose of that after the procedure. So I have a slide out, and I have some slide covers as well. We are going to go ahead and just write our patient's name on here, which is Finn Brooks. And then what we're going to do is we're going to go ahead and get some of our methylene blue stain, and we're going to place that into our, just one drop into our tube. And this is a clean pipette, by the way. So again, I'm going to do one single drop into the tube. And then we're going to dispose of that. And then we're going to use the pipette, and you can see how the stain isn't completely mixed in with the sediment. So we're going to use our pipette to just mix it in, and we can kind of draw it up and push it down, and draw it up and push it down, and do a good mixture there. There's a few bubbles, so we'll kind of smush them back into the solution, but basically we just want this mixed nicely, and then we're going to go ahead and draw it up again. And so what we're going to do is we have a nice little bit in the end here. We're going to place this onto our slide. We only need one drop. And then we can just rest that in there. And then we're going to place our cover slide. And so what we need to do is we need to go ahead and take a look at this under the microscope. And so I'm going to, since my microscope has not been touched by anything with chemicals on it, in order to prevent my microscope from getting, because I have formalin on my gloves and I don't really want to touch the microscope, I'm going to go ahead and take my gloves off and put new gloves on without touching my skin. That will just make me feel better when I go to disinfect everything, that I'm not just like touching everything with contaminated gloves. So I'm putting clean gloves on. I'm going to go ahead and carefully remove my goggles, and then we're going to go ahead and examine this. For this, we need to examine it just under the scanning lens. We should not need to go up to the higher magnification to identify the microflaria. And so I just need to focus this in. Alright, and as usual, I am going to follow a very systematic way of looking at this. We need to make sure that we're not missing large areas, so I'm going to start out at one corner and move up and back in a very systematic way. As I'm looking in here, I can see many blood cells, which is normal. they're sort of dispersed throughout, so it's sort of like peppered on here, and like if I had taken a blood sample that was a red blood sample and put it on here, it would be so many red blood cells that I couldn't read it. They would just be like, I mean, like when you do a direct smear, so you have just tons and tons of red blood cells, and you can't see between them. It's like a whole uniform mesh of red blood cells. So that's not what this one looks like. It's sort of like peppered out through here, but there's still, I mean, plenty of them. So I'm at the corner, and I'm just gonna start reading all the way down. I'm going to adjust my little iris diaphragm really quick. And so with this modified Knots test, it's a little bit different than a direct smear, because the microfilaria are going to not be moving in this sample. So if I see one, it is not going to be moving. And so it's a little bit different. You know, I don't want to read it super fast and miss something, but everything I'm seeing is very like they're all little blood cells. There's not a lot of like debris on here. There's a couple of like clumping and things like that, but it's not like an ear smear where it would be a lot of debris or something like that. So I will just keep on scanning. And looking for anything that doesn't sort of fit with everything else I'm seeing. So with this dog, it is fairly unlikely that I'm going to see anything. This dog is on monthly heartworm prevention, as they should be, but this is part of their annual heartworm screening. We're doing a general annual heartworm test. We do the SNAP 40X, and then we are adding this on as a secondary check. The American Heartworm Society does recommend that dogs get both a heartworm antigen test and a heartworm microfilaria screening once a year. So ideally we would do this with every patient. That's certainly not standard at this point in time, because it is time consuming, and a lot of owners might not want to pay for both tests financially, but that is what the American Heartworm Society recommends. So we are going to go ahead and do that with this patient. But even though it's unlikely that I'll see anything, I do have some pictures printed out for us so we can discuss what it would look like if we did see something. But it's still great practice to do both types of screenings. I think we're going to have about one more up and down. I'm doing the up right now, and then we should be at the end of the slide. Alright, so I've reached the end of the slide. We can go ahead and turn off our microscope. So I didn't see anything. So we would call this, for the test results, we would say no microfilaria seen. If we had seen an organism that looked like microfilaria, we could not just say, oh, we have microfilaria. So there are the Dirofilaria immitis that were actually pathogenic larval heartworm, the larval stage of the heartworm. And then there's another non-pathogenic nematode, which means it doesn't hurt the dog, it doesn't do anything that we need to treat for, which I'm going to preface this with, I probably am not pronouncing this correctly, and I apologize for that. I've only seen it in writing, and I'm bad at pronouncing things in general. So the nematode that we're looking for is Dipetalonema. I don't know if I said that right. Reconditum. So Dipetalonema reconditum is different than Dirofilaria immitis, but they look very similar. They're both sort of little round worm things that would be in there. So I have some pictures that we're going to talk about. I have two different photos. Well, they're the same photo. These are just cut out papers. So they're the same photo, but I have two different things to talk about, and this photo is a little bit clearer. So this is a photo of a comparison of the two organisms. The Dirofilaria is the one on the bottom, so that's the one that we are worried about being pathogenic. The Dipetalonema is on the top. So the reason I have this one is because I really like the way that they describe this in this photo. So one thing is that when you're looking at the slide, you probably aren't going to have both of these organisms that are sitting really nice right next to each other for you to compare and say, oh, well, this one's microfilaria, this one's not. So if you look closely, and what this text says to kind of show you, but if you look at the width of the parasite and you compare that to the blood cells, our Dirofilaria are actually wider, or just kind of, they're like as wide or a little bit wider than the blood cell, whereas the Dipetalonema is it's thinner. So that's like a really, that one just resonates with me as a not super experienced I don't want to say pathologist, that's not right, but anyway, I don't read these a lot, and I don't do microscope work every single day. I don't work in a lab where this is done over and over again. So that is something that is, I can really grasp that idea. But anyway, so this is another chart here that it compares the two organisms. So basically, other things that we're looking at between the two organisms is that the Dirofilaria are both longer and wider than the Dipetalonema. So this little chart here shows you length and width. When we're reading the length and width, or like on the microscope when we're reading it, we're reading it in micrometers, so very, very small, but you can see the lengths are listed here. For the dirofilaria, it's an average of about 310 micrometers. This doesn't seem like a big difference when they're like so tiny, but the average for the dipetalenema is about 280 micrometers, so there is a difference between those two. Width is also, again, it's close, but when you're dealing with such small units, that's why it seems a little bit not impressive. But for the dirofilaria, again, we're looking at about 7 micrometers. For the dipetalenema, we're looking at like 5 micrometers. So, and you can see that one's, I think, a little bit more obvious when you look at the picture, the width of them. But again, if you're not looking at both of them at the same time, that's not something that would be as obvious. So, it's also some body shape differences between the two organisms. So when we look at the shape of the head of the organism, which these two are facing different directions, the head of the dirofilaria is tapered. The head of the dipetalenema is a little bit more blunt. That's a very subtle difference. It's sort of hard to see that. You kind of have to look at like not just the very tip of the head, but kind of the first little eighth of the organism, where this has a very slight little taper, and the dipetalenema does not. There's also some difference in cellularity of the anterior end of the organism. So again, if we look at the anterior, can't point right because I'm backwards, but so if we really look at this anterior end of the dirofilaria, you can see that there's sort of some clear space in there. And then if we look, oh, and this picture is cut off, so that's why, again, another reason I have this one. If we look at the end here of the dipetalenema, we can see that it's pretty much blue and cellular all the way down to the end. So again, that's pretty subtle, but it is there. Like you can obviously see it when you compare the two pictures. Finally, we have a bit of a difference in the body shape and the tail shape. So typically the dirofilaria is going to be straight. Its body is going to be straight. Its tail is going to be fairly straight. And you can see that in this photo. When we look at the dipetalenema, and I'll get the other photo out again because this one's not cut off. The dipetalenema tend to be, their overall body shape tends to be a bit more curved. And then the tail shape is described as a button hook. So it's got like a little bit more of a hook to it than the straight tail of the dirofilaria. So that's what we're looking for. That's the main differences. So, you know, those are things that we would need to notate, and if we did see an organism, we would need to take some time and really notate those subtle things to make sure that we're giving a proper reading of the microscope. So I'm going to go ahead and properly disinfect and dispose of all of our samples. I am going to go ahead and put my safety goggles back on to do that, and I have my gloves on, so I'm going to go ahead and take care of all of that, and then we'll notate the record, notate the result in the record, and that is task number 8.